A great good morning to all. As everyone knows, myself, Dr. Jyoti. Today I'm here to discussing with you about the topic Introduction, History and Scope of Biotechnology. As you people know, we have the uh, fundamental techniques of biotechnology, journal history, histology, and cell tissue culture, and preparation of cell culture media, the stem cell, and propagation and maintenance of tissue culture, preparation of cell lines, cellular aspects of tumor cells, in vitro fertilization and embryo transfer. All these comes under block 1, and as a part of block 2, we have applied biotechnology, which is going to cover the units like 9 to 16. In that, we are going to studying about the agricultural biotechnology, aquatic biotechnology, microbial biotechnology, environmental biotechnology. As a part of environmental biotechnology, we are going to cover the environmentally important microorganisms, wastewater treatment, and wastewater utilization. In the environmental biotechnology, we are going to covering the topics like landfill, composting, earthworm treatment, and degradation of plastic. Along with that, uh, bioremediation, enzyme biotechnology, and industrial biotechnology. Actually, we have 16 uh, units as a part of block 1 and block 2. So we'll do one thing. As we are having these many classes, we will cover two topics, two units in one class. Then we will cover all the topics. So today we'll discuss about the topic like introduction, history, and scope of biotechnology, along with the journal histology and cell tissue culture. That, as everyone knowing, animal biotechnology. Animal biotechnology is the combination of science, uh, history, scope, and significance of biotechnology. In this animal biotechnology, the animal biotechnology is the combination of science and the engineering, which is very useful to transforming the living organisms. The main objective of animal biotechnology is to make the products and to improve the animals and to develop microorganisms for specific agricultural needs. And that creating the transgenic animals is the best example of animal biotechnology. So the biotechnology which is useful or which use of artificial methods to modify the genetic material of living organisms or cells to produce the novel compounds or to perform new functions. Here, the root words of biotechnology is long back, like ancient one. So the meaning of this bios means life, technicos means skillfully made tools, and the logos that is a word or the essence. So here the biotechnology means the life of living tools. It is used in different areas like as just we discussed, agricultural processing, agriculture, and industrial production, and environmental cleanup, and medicine production like novel compounds identification, enhance the crop yield. So the biotechnology that has been used for improving the livestock and the crops since the beginning of the agriculture through the selective breeding methods. So the Since the discovery of the structure of DNA in the year of 1953, and principally after the development of methods to manipulate the DNA, the year of 1970, the biotechnology is equally significant to manipulate the organisms, DNA at the molecular level. So here a set of a modern tools that utilize living organisms or the parts of it, cell or tissue or gene to make or modify or improve the plants or animals or develop microorganisms for the use and their large scale production. So here this technology has key applications in the medicine for the production of vaccines, antibiotics, and in agriculture for the genetic modification of the crops. In addition, the biotechnology has many industrial applications like fermentation and the treatment of oil spill and the production of biofuels, as well as many household applications like the enzymes which is used in the laundry detergents. So here the genetic engineering, also called genetic modification or gene manipulation, the direct manipulation of the organism gene using the biotechnology is a set of technology used to change genetic makeup of cells, transfer of within and across the species, that boundaries to produce the improved or novel organisms. The new DNA that is obtained by either isolating or copying the genetic material of the interest which is used using the recombinant DNA technology, that molecule which is made by the Paul Berg in the year of 1970 by combining the DNA from the monkey virus, that is the SV40, with the lambda virus, as well as the interesting genes. The process that can be removed or knock out the genes, the DNA that can be inserted randomly or target to a specific part of the gene. So here the utilization of microorganisms or organelles, cell organelles, or the biological process to the mankind for solving many problems for the welfare of the mankind. The convention of biotechnological or biological diversity 2000, the biotechnological and application that uses biological process or biological systems and living materials derived to make or modify different types of products. And the biotechnology, here the production of goods and services. And yes, definitely it is the biology that have many problem solving equations make use the most prominent area of biotechnology, the production of proteins and drugs, as just we discussed. And along with this, yes, gene therapy is before going to the basic techniques of the biotechnology, like tissue culture and genetic engineering, as we have all these in our unit 2. We wanted to discuss, I wanted to discuss about some topics, journal topics like the gene therapy is the medical field which focuses on the gene modification of cells to produce a therapeutic effect or the treatment of disease by repairing or reconstructing the defective genetic material So that here the first attempt at modifying the human DNA was made by or performed in the year of 1980 by Martin Klein. But the first successful nuclear gene transfer in the human by NIH, National Institutes of Health, uh, was performed in the year of 1989. This the first therapeutic use of gene transfer as well as the first direct insertion of human DNA into the nuclear genome that is performed by the scientist French Anderson uh, in a trial which starting from the year of 1990. Though this is to be enabled to cure many genetic disorders or treat them over the time, uh, there are biotechnological basic principle of genetic engineering in animal systems uh, that are vectors. As just a slide saying that, yes, we have two types of techniques that is used in the biotechnology are tissue culture. We call it as soft biotechnology, and the genetic engineering, hard biotechnology, as just we discussed, genetic engineering, genetic. So this is called the hard biotechnology. So the systems that are uh, used in the genetic engineering, those were like vectors. Here the vectors like plasmid, cosmid, phasemid, and yak means yeast artificial chromosome and the bacterial artificial chromosome. We also, in short word, we call it as a BAC. And shuttle vectors, yeast vectors, mini chromosomes, and the gene transfer methods in the animals are electroporation, microinjection, biolistic particle delivery system, and sonoporation, and the preparation of media and sterilization technique. Here we use the natural and synthetic media, and we use different types of cultural methods, and we also have different types of uh, microorganism identified methods like hanging and characterization of transcontinents. And we have some scaling of technologies like bioreactors, transgenic and xenotransplantation, and artificial insemination, and embryo transfer, and isolation and purification of nucleic acids, and hybridization techniques through southern blotting and western blotting and northern hybridization techniques. Along with that, we also have uh, the techniques like PCR, RFLP, RAPD. Those were the markers. Along with that, we have DNA fingerprinting, DNA barcoding, and Maxon and Gilbert methods, and Sanderson method, and automated DNA sequencing methods, pheromones identification, and knockout and knock-in technology. Along with that, we have gene therapy and human genome project and ethical issues in the genetic engineering and transgenics. Uh, transgenics. All these we study by using the biotechnology. Here the applications of scientific and engineering principles of processing of materials by biological agents to provide uh, services. Here the basic techniques that is depending upon the nucleic acid, which are macromolecule made of nucleotides and nucleosides. The biotechnology that can be defined as the technology based on the biology. Here, the term that includes the cellular and biomolecular processes to develop the technologies and the products that assist and advance our life and health. The applications of biological processes of microorganisms for more than 6,000 years make useful food products. Those were bread, cheese, and the preserved dairy products like the origin of biotechnology. That is, it's a old as the human civilization science. It is a little more than 100 years old. Fermentation, not 100 years more, as it's like 6,000 years back. Fermentation, which is a oldest biotechnological process that discover people by prolonged soaking of grains or sorting juices and other processes practiced by the man, including the laying and for the production of alcoholic beverages, production of vinegar, and production of alcohol. In the modern biotechnology that provides the cutting edges products and technologies to combat and debilitating and rare diseases that reduce our environmental footprint, feed the hungry, use less and cleaner energy, and have safer, and cleaner and more efficient and actually processes. At present, nowadays, there are more than 250 biotechnology healthcare products, and the vaccines, those were available to the patients largely for untreatable diseases. More than 13.3 billion of farmers, those around the world, using the agricultural biotechnology to raise the yield and reduce the damage from pests and farming impact on the environment. The recent trends and advances in biotechnology are helping us to get ready for meet the society most imperative challenges. With this here, as everyone knowing, the hero of biotechnology, like the term biotechnology, which is coined by the Hungarian engineer, he is Karl Erke, in the year of 1917, to describe the process for large-scale production of pigs. According to him, every jobs are biotechnology by which the products are produced from the raw material, which using the living organisms. At the conclusion of the 20th century, biotechnology that is emerged as a new dimensions of the biotechnology, amalgamated with the technology. There was no rapid sprout of this discipline, but some methods were developed centuries back. Therefore, in the nutshell, the biotechnology is concerned with the utilization of the biotechnological concepts or components for the creation of useful products. As a side saying that 6000 BC, the Sumerians and Babylonians knew the concept of the wheat production, After that, Aryan, they started preparation of curd. In the same era, the Greeks, those were probably first to prepare the cheese. It is the first fermented food. As the father of microbiology and father of industrial biotechnology. He, the fellow who served for the biotechnology for 1822 to 1895, and in that time, he's the one who discovered the yeast for the production of the alcohol, and he's the one who identified the bacilli, rod-shaped bacteria for the production of the acetic acid, and he is the fellow who said the storing of wine is by storing of wine, which is by rod-shaped bacteria, bacilli. As everyone knowing that Clostridium acetobotanicum, he employed the Clostridium acetobotanicum. He observed the Clostridium acetobotanicum for the production of butyric acid and acetic acid along with the alcohol. After his contributions, in the 1929, Alexander Fleming, he discovered antibiotic penicillin, and in 1973, first successful genetic experiments were carried out in famous world laboratories. And so here the history of biotechnology comes like as just we discussed about the term of biotechnology. Thus, the biotechnology has broadly defined as the development and the employment of biological process, forms, and the systems to obtain the greatest benefits to the man and the mankind in the other forms of life, in order to make bread, alcohol, beverages, and cheese, preservatives, and the dairy products. But such processes are not what is meant today by the biotech. So the biotechnology field that is led by the Genentech, a pharmaceutical company, and Boyer and Paulberg and Stanley Cohen. Those were the pioneers for identifying the recombinant DNA technology. We already discussed. So, when coming to the as just we discussed here, the classical biotechnology and advanced biotechnology. When the human civilization started, the development of biotechnology started. That can be discovered two growth phases like classical biotechnology and advanced biotechnology. Here, the classical biotechnology is traditional biotechnology, which was developed by our ancestors, like fermentation bacteria, such as animal fat, ghee, which is fermented of milk. Similarly, the preparation of curd, wine, beer, vinegar, all these. In Indian Ayurveda, the production of asava and arishta is done by using the substrates like the flowers of Maduka indica and is that Maduka indica only, which is characterized till today, since Vedic periods. So in all these materials, various substrates are transformed into a number of products. So then coming to the advanced biotechnology, all these we just discussed, like recombinant DNA technology, the technology which now we are enjoying, like for more than a decade, the biotechnology industry was dominated by the rDNA technology or the genetic engineering. This technique, which consisting of splicing the genes and for the useful production of protein, uh, often a human protein, into the production of cells such as yeast and bacteria or mammalian cells in the culture, which then began to produce the protein in the increased volume. In the processing of splicing a gene into a production cell, a new organism is created. At the first, the biotechnology investors and researchers are uncertain about whether the courts would permit them to acquire the patents or organisms. After all, patents were not allowed on new organisms. That is what happened to the discovered and identified the nature. As everyone, every biologist familiar about the Anand Chakravarti's case. So he's the fellow. He identified the microorganism called Pseudomonas putida. When he went to climbing his bacteria, uh, see, in the year of 1980, that case, he suited the case against the US government for they said that he live human-made organisms patentable subject matter. This decision which spawned a wave of new biotechnology firms, and that infant industry first investment in the 1982, the recombinant insulin became the first product which made through the genetic engineering secured approval from the US Food and Drug Administration FDA. Since then, dozens of genetically engineered protein modifications that have been commercialized around the world, that including the recombinant versions of growth hormone, clotting factors, proteins for stimulating the production of red and white blood cells, interferon, and clot dissolving uh, agents, and the possession or ownership of the technology and its socio-political impact. At the moment, the conventional industries and the pharmaceutical industries uh, and agro industries are targeting their attention to obtain the biotechnology-based products, the new or modern biotechnology that embrace the methods of genetic modification and cell fusion and fermentation technology uh, to find the progressively or more and wide industrial applications. So here the biotechnology is a interdisciplinary science the key difference between the biotechnology and the biotechnology is their scale of operation. Usually, the biotechnologist, the one who works in the range of nanograms to milligrams. In our group, most of the people are knowing about the biological materials. Like if someone wanted to purify a compound or purify a new product uh, from the bulk, like if and biotechnologists wanted to purify a specific compound, drug of choice from the plant material, he has he required a cages of plant material. Then only he will get like in more than in uh, worst cases, he'll get 5 grams of compound. Uh, so for scaling up, uh, we have the biological process, we have the biotechnological process for scaling up the compound after the identification. But a biotechnologist, uh, he knows the range of the material. Definitely, he will get in the nanograms or in the milligrams. However, the biotechnologist working on the production of vaccine may be satisfied with the milligrams of yield. Yes, obviously. Why? Because many other projects, they will aims at the kilograms or the tons, but they'll get a milligrams of yield. So, but definitely, he's the one who will satisfy with that milligram. That's the main objective of biotechnologies. It is consisting of the scaling up of the biological processes. The scientist who is Smith in the year of 1996 for large-scale cultivation microbes and the cell and their upstream and downstream processing. These processes, usually the biotechnological processes that involves like strain selection and improvement and mass culture and the optimization of cell responses and a process operation and downstream processing. And in the downstream processing only, we will call it as like product recovery. So it is just like a 5-step process. So this 5-step major operation that mainly depending upon the product quantity and the quality. So here the many areas of biotechnology, the amalgamation of biology with the technology, the term of biotechnology, this field that utilizes like biology and engineering and biomolecular processes and biochemistry, biophysics, cell biology, and colloid chemistry and embryology, ecology, genetics, immunology, molecular biology, and medical chemistry, pharmacology, polymer chemistry, and thermochemistry, and virology. Here, from the basic biology to the improvement of new products with the different types of technology and the process that facilitated by the biotechnology. As just we discussed bread and preserved dairy products like cheese. Here it is the technology, biotechnology, which positively impacts the several fields such as agriculture and food processing engineering or food processing and physics like energy and environmental factors. So it is the science which plays an extremely crucial role in the development of medicines and the vaccines. The one who are the witness for COVID-19, they know the taste of biotechnology, how biotechnology people worked on for the production of vaccines. So we are here today discussing about biotechnology. Why? Because we are the witness for the COVID-19. So here, the biotechnology that covers the areas like biopharmaceuticals, pharmacogenomics, and genetics. Uh, Extensively use the biotech uh, processes. Uh, like this enables the methods to discover and cure the life-threatening diseases. As everyone knowing, human insulin, and which is used to treat the diabetes, is made in genetically engineered bacteria through the process like biotechnology. Here it is an interdisciplinary subject which links to the microbiology, genetics, biochemistry, chemistry, food science, food technology and engineering, mechanical engineering, and chemical engineering, biochemical engineering, and electronics. As we already discussed, yes, in the past, uh, biotechnology which eradicated the viruses like SARS, influenza, Zika, and yes, the reason why biotechnology companies and the research organizations are optimistic for COVID-19 vaccine as well. So this uh, now the entire biotechnology industry is going through a wave of evolution. This evolution will definitely bring new job opportunities and research opportunities for the students, that hence redefining the scope of the biotechnology. So in our India, we have the after the global applications of the RDNA technology, the government of India that has set uh, an official agency called the National Biotechnology Board. We also called it as like NBTB, the one which started and functioning under the Department of Biotechnology, DST, in the year of 1986. Here, the National Biotechnology Board that was replaced with a full-fledged department, that is familiar, like the DB, the Department of Biotechnology. Under the Ministry of Sciences and the Technology of Planning, promotion and coordination of various programs. At present, in India, we have like DBT, DST, CSIR, ICAR, ICAR, ICMR, ICGP, and IAR, and some other agencies also we have. That this sequence of programs or departments which is working under the government, these agencies and the other national and industrial and international manufacturing biotech products and marketing them after the clinical trials. Technology Development Board, that is the Technology Development Board, TDB, that has been set up by the government of India for the promotion of products that are developed and tested. The Technology Information and Forecasting and Assessment Council, that is the TFAC, have prepared a Vision 2020 document which consisting of the biotechnology. That also, since 1980s, India has supported a lot to the biotechnology industry and its end products and its products. So that currently there are over 30 companies in India which are producing the modern biotech products. Here it is a multidisciplinary pursuit that emerged as a demanding and besides being a branch of advanced biological sciences, it has attracted many multinational companies, including concerned with the production of pharmaceutical products for the cure or control of many human diseases. These products, including the antibiotics, vaccines, life-saving drugs, and gene therapy, and the implement of clinical testing diagnostic tools, and the production of novel varieties of crop plants uh, and animals. So, as just we discussed, after the Watson and Crick experiments, after identifying the DNA, the unified definition of the genetic engineering that has been given by the Smith, the formation of the new combinations of heritable uh, material by the insertion of nucleic acid molecules that produced by like whatever it means outside the cell into the any virus after duplication, bacteria, plasmid, or other vector system as to allow their incorporation into the host organism in which they do not naturally occur, but in which they are capable of continued propagation. After 1970s, there are many identifications as just we discussed. Genentech is the US company in the year of 1978. They produced genetically engineered, they used genetic engineering as a technique and a tool to produce human insulin by employing the bacteria called E. coli, Escherichia coli. And in 1996, the first cloned lamb, Dolly, that was born successfully by the efforts of scientists of Scotland. Thereafter, several cloned animals were produced in the year of 2001, and the sequence of human genome that is completed by uh, the year March 2003. And on December 27th, 2002, claim was made for the birth of clone baby called Eve. Uh, most of the biotechnologists know the name of Dolly because as it is the first baby of biotechnology. That is why since 1996, everyone knowing that Dolly is the first baby of biotechnology. So the second baby in the 2002 is a clone baby that is Eve by the scientists by Human Cloning Society, clonide of France. In 2005, the scientists of South Korea that have used a method called therapeutic cloning to produce the stem cell lines. In this method, human embryos were produced through cloning, and the stem cells were obtained from the blastocyst. The existed stem cell that could be grown uh, in vitro, and they used it for further use. So, other field like bioinformatics is a hybrid science that links biological data, uh, where uh, the techniques for information and storage and distribution and analysis to support the multiple areas of scientific research, where it includes the biomedicine and bioinformatics that fed by the highly throughput data engineering experiment that includes genomic sequence determinations and measurements of gene expression patterns. The database projects curate and annotate the data and then distribute it through the World Wide Web, www. Mining these data leads to scientific discoveries and the identification of new clinical applications. In the field of medicine, in particular, a number of important applications for bioinformatics that have been discovered. We have a couple of examples for this. It is used to identify the correlations between the gene sequences and diseases to predict the protein structures from the amino acid sequence to aid the design of new drugs, like novel drugs, and to tailor the treatments to individual patients depending upon their DNA sequence. The entire processing is called like pharmacogenomics. So, applications like as just we already discussed, like about the human insulin. Yes, definitely we can say like we have one example for the ancient biotechnology, like Pandavas and Kauravas story. So Draupadi, because of her angry, she broken down her pregnancy and made pregnancy in different 100 pieces. So, as the instructions made by her brother, the Shakuni, he kept all the pieces in different pots, employed some ancient biotechnological applications. Surprisingly, the story you people know. So we can say like all those were the pioneer steps for the biotechnology development. So there are many, not only this story, this ancient story. I couldn't say like story. It is, yes, it is one of the examples. Biotechnology is there in the ancient India. So the natural production of the modern biotechnology includes the scientific methods. Those were genetic engineering that have enhanced the natural capabilities of natural production of organisms, bacteria like E. coli, and producing the mammalian hormones. Those were insulin, somatostatin, somatotropin, and yeast cells that have been genetically manipulated for the production of vaccines against the hepatitis B virus and myeloma cells, like cancerous cells of bone marrow, and the beta cells, like B cells of immunized mice, were hybridized to production of the hybrid cells that consisted characteristics of both the cells which were having the cell division and antibody production. Now the hybrid cells like hybridoma. You people know about the hybridoma technology, and those were being used for the production of monoclonal antibodies that following are different biotechnological tools. There are maximum number of benefits of biotechnology that have been utilized by healthcare industry. So the biotechnology that derived the protein polypeptides from the new classes of potential drugs like humulin, human insulin, Recombivax, HB, hepatitis B vaccine, and some problems like nutritionally influenced diseases. Those were diabetes, arthritis. So even for that also, biotechnology provided the valuable and viable alternatives for the food problems. So the major landmark in the human history is the production of human genome sequence. The Human Genome Project is an international research program. Almost the whole human genome that have been sequenced and chromosome mapping has been developed in various laboratories throughout the world that coordinated the human chromosome mapping that was completed in the year of 2003, March 2nd. And about 33,000 functional genes in the human is identified. And more than 97% of the genes are non-functional. They do not encode for any polypeptide chain. Here, the main objectives for the Human Genome Project that the construction and detailing of genetic and the physical map of human genome. To determine the complete nucleotide sequence of human DNA, to store the information in the database to locate the estimated genes like 50,000 to 1 lakh genes within the human genome to address the ethical, legal, and social issues that may arise from the Human Genome Project, and to perform the similar analysis on the genomes of several other organisms. Natural biodegradability of pollution that is presented in the environment is definitely increasing. So with the use of biotechnology, the bioremediation technologies have been found successfully to combat the pollution problems. The bioremediation is the use of microorganisms to detoxify the pollutants present in the environment, usually in the soil and in the water or water sediment. The pollutants that cause several health problems. The microorganisms to show the potential or degradation of oil, pesticides, and fertilizers that belong to the genera of bacteria. Those were like Pseudomonas, Micrococcus, Bacillus, and fungi. In fungi, Candida, Cladosporium, uh, Torulopsis, and Trichoderma. Uh, yes, we also discussed about the genomics, computational, computer-based uh, sequencing methods like bioinformatics, and we also discussed about the mathematics and uh, statistics and the computer science for understanding of biotechnology, biochemistry, uh, biological data, the remarkable success of bioinformatics, the data that has use of shotgun sequencing of human gene project. So the biotechnology, the production of interaction between the biological sciences and modern technology. During the last 2 decades, the biotechnology has achieved the greatest advancement in the field of recombinant DNA technology and polymerase chain reaction, gene cloning, DNA fingerprinting techniques, being a branch of advanced biological sciences. It has already attracted many multinational companies. Yes, industrial insulin production and the production of growth hormones is one of the greatest blessings of biotech. This is the introduction and scope and significance of biotechnology. Then we'll start another topic. We'll start another topic called animal tissue culture and the basics of cell culture. So hope you people understood about the ancient biotechnology and the modern biotechnology. And we also discussed about the introduction, history, and the scope of biotechnology. So after that, we have the topic called animal tissue culture and cell tissue culture. Before going to the cell tissue culture, we have to know what is cell and what is cell line and what is tissue. So we will briefly discuss about animal cell culture. Then we'll go with the so here animal tissue culture is the growth of tissues that separated from the animal in vivo and in vitro. These two are the terms in vivo or in vivo. Some people used to pronounce it as a in vivo, and some people used to pronounce it as a in vivo. So means within the animal, within the living organism. So in vitro means within the laboratory. So whichever the techniques we are used for enhancing the or speed up the reaction after using the laboratory techniques are called as in vitro. So here we are reading the topic like animal tissue culture is the growth of tissues that is separated from the animal origin or animal body, uh, tissues that are separated from the animal and they are growing in vitro, means within the lab. So when coming to the term like cell line means every cell that is presented in the human body is not capable of growing in lab. Only a few types of cells that are we can able to grow in vitro. So, but they are neither suitable for industrial use nor for the scientific purposes. We'll see why. Why means because many cells that die during the course of time that releasing the toxic substances which inhibit the activity of other living cells. So one cell that inhibit the growth of other cell. In order to avoid this type of problem, to achieve an exponential cell growth, the cells are converted into immortal. Means though it is having the life, but it can't able to function itself. So called as immortal cells. Such immortal cells are called as cell lines. So yes, there will be a procedure for production of cell lines. So what we have to do? We have to take a piece of tissue which is removed from the organism, whichever it may be the organism, whether it is the human body or it is a pig or it is a rabbit or it is anything removed from an organism that is grown in the laboratory. That adhesion of between the cells is broken with the enzymes like by using the enzymes like trypsin or collagen. I will show you the protocol in the next coming slide. Then you will definitely understand the procedure. No worries. So the cells that are transferred to a plastic dish or a bottle which containing the culture medium. Here you can see the procedure. Just now I told you, no, here it is a tissue. The tissue which is removed. The tissue which is removed from the organism and they treated with the trypsin or collagen. Then after the trypsinization, the process is called as trypsinization or the collagenization. This tissue is becomes like the separated cell. So these separated cells that are transferred to a plastic bottle or plastic dish. Here the plastic dish which containing the culture media. So here it is a culture containing plastic bottle. So it is incubated at the temperature 37 degrees with 5% CO2 and with 90 to 95% O2. So the cells which is growing within the surface of the container, this culture is referred to as the primary cell culture. All the cells will stop dividing due to the contact inhibition. So the cells are transferred to the once the primary cells means the cells which is reached to a optimized growth. optimum growth, not optimized, optimum growth, the cells is separated and transferred to the fresh medium. After reaching to a optimum growth means when the contact inhibition started about 50 dilutions, there is no growth of culture. So this is telling us that the cell sorting means cytotoxin production got started. So here the diploid cell strain, the surviving cells, those were transferred to the culture media, means new culture media. Then the cell growth started. We have to repeat these stage about repeated dilutions where cell sorting started. Here you can see the cells with a heteroploid cell. Then about after 50 dilutions, you can see the presence of cell lines. This is how cell lines may get developed. So the some cells continuously grow, such as diploid cell strains. The cells which is reached to the some amount of the passages, then after 50 passages, they lose the ability to grow. A few cells among the diploid cell strain will survive. These are termed as heteroploid cells because they may undergo many chromosomal rearrangement and deletion. These cells will grow indefinitely as long as the medium is replaced, becoming effectively immortal. These survivors are now calling as cell line. So what is the culture medium here we use means here it is a supportive media, supportive environment which creates a natural environment. It is the environment that provides for the growth of uh, the cells in the laboratory, similar to those conditions that our cells have been exposed to in vitro. Here the culture media is consisting of a physical media, a support or the matrix, and the chemical media that is having the appropriate nutrients and hormones and all the stromal factors. And the serum, the one which plays the major role, uh, since it is the most economical, easily available, and most widely used in the culture medium for the animal cell culture. Fetal calf serum, which is the preferred one. The major functions of a serum as a culture medium to provide the nutrients, hormones, growth factors, and the attachment and spreading factors, binding proteins, vitamins, minerals, and lipids, a protease inhibitor, and the pH buffers. So here you can see the media bottle. This is how a media bottle looks like. So as just we discussed about the serum, there are some disadvantages of serum because yes, there might be a chance of contamination. Serum definitely easily contaminated by the virus, fungi, and the bacteria. Some enzymes that are presented in the serum can convert the cell secretions into the toxic substances. Now we have artificial culture media uh, like serum, serum-free media, protein-free media. Those were like EM media. Here EM, it stands for Eagles Minimal Essential Medium. And EM media, Dalbecos, modified, enriched media. And RPMI, Roswell Park Memorial Institute. These are the best media. Nowadays, many industries and many laboratories, many researchers are widely using only these three media. Those were EM media, DM media, and RPMI media. So, what are the types of cell culture? Yes, we have the primary cell culture, the maintenance of the growth of cells that are separated from the parental tissue in culture media using the suitable glass or plastic container is called as primary cell culture. Just now we have discussed in the past slides. I have also shown you the method or the steps that are involved in the cell culture. So here it is the primary cell culture, which is separated from the tissue, which is separated from the parental tissue in the that is growing in the culture media. And monolayer culture, this is in turn also called as adherent cells. The cells shown to require attachment for the growth. They are usually derived from the tissues of organs such as kidney. And the suspension culture, the cells which do not require attachment for the growth, they are derived from the cells of blood system. So here you can see the entire process. The cells that are anchored means attached surface and divided. And when the cells have formed a complete single layer, monolayer, they stop dividing. Here it is a density dependent inhibition. If some cells are scraped away, the remaining cells are divided to fill the gap, then stop because it is a density dependent inhibition. So, what are the advantages in propagation of cells by suspension culture methods? Obviously, it is a faster method. The frequent replacement of medium is not required. So, this have a short lag period. You people know what is lag period, lag, log, and stationary. So, here the cells which having the very short lag phase, when the all the nutritional requirements are filled, it will definitely directly goes into the log. So the treatment with the trypsin is not required in the suspension cultures as the homogeneous suspension of cells is obtained. The maintenance of them easily and bulk production of cells is very easily achieved. And scale up is also very suitable. Here this is how the suspension cells looks like. And the secondary cell cultures are the cell lines. When a primary culture is subcultured, it becomes unknown as the secondary culture or the cell line. So the subculture means passage. I already discussed with you. Even in the physical classes also we discussed. Passage is the transfer of cells from one culture via to the another culture vessel. There are two types of cell lines or the cell strengths. Those were finite cell lines and continuous cell lines. Here, finite means have a limited lifespan. On the other hand, continuous cell lines have unlimited lifespan, which exhibit the heterogeneity, and they grow in monolayer. Continuous cell lines grow in monolayer or a suspension form. Finite cell lines that exhibit the property of contact inhibition. Here, the continuous cell lines don't have any contact inhibition. The growth rate is slow and the growth rate is rapid. Doubling time is 24 to 96 hours. But here in the continuous cell lines, in another way, we are adding the media continuously. Because of that, the doubling time is very less, 12 to 24. So how to scale up the animal cell culture? Scaling up is modifying a laboratory procedure so that it can be used as an industrial scale. So here we have the roller bottles. I will show you what is roller bottle and which is having the beads uh, for removing the adherent cells. The roller bottles provide a total curved surface area of micro carrier beads for growth. The continuous rotation of the bottles helps to provide the medium to the entire cell mono layer in the culture. So this system have some advantages like this increase the surface area. This provides the constant gentle agitation that provides the cells to utilize the medium, and the one which provides increasing ratio of surface area of medium to its volume. You can see this is how a roller bottle looks like. So just you can consider this is the cells and this is the media and continuously wheeling. And microcarrier beads. So here the use of microcarrier beads is these are small spherical particles which is having 90 to 300 millimeter size, which is made up of by the dextran or the glass that increase the number of adherent cells for the flask, and the range of densities and size. The cells that grow at a very high density, which rapidly uses the medium at the recommended concentration. When the microcarriers are suspended, they provide 0.24 meter square area for every 100 ml of culture flask. This is how the vials looks like. So the roller bottles looks like this, and the microcarrier systems in the bioreactors during the virus production and M.sc cell growth. From this downstreaming process starts, like here the other steps like clarification, inactivation, and cross-flow filtration, finally chromatography. So what is the applications of animal cell culture? They are used as a substitute host to study the pattern of viral infection, and they are used in the manufacturing of vaccines, antibiotics, antibodies, and hormones, interferons, vitamins, steroids, and pharmaceutical drugs. They are the good tools for testing the potency of drugs. They are served as the model to study the metabolism of various substances. They are used in the study of effects of toxins and the contaminants in the cancer research, which requires the study of uncontrolled cell division in the cultures used in cell fusion. So these are the examples. So when coming to the cell culture, is the process by which prokaryotic or eukaryotic or plant cells are grown under controlled condition. But in practice, it refers to culturing of cells derived from the animal cells. The cell culture, which is identified firstly by the Ross Harrison in 1907, Ross in 1885. He is the one who first time maintained the embryonic chick cell in a cell culture. Here you can see the Ross bottles. I'm sorry, Geeta. Just now I have seen your message. I'm already sharing the screen. Oh, sorry. Why it got paused? I don't.
It is not coming, ma'am. Not visible.
Right? I don't know. It is showing that your screen sharing is paused.
Not yet, ma'am.
Sorry. Geeta, am I audible?
Yes, ma'am.
Ah, sure. There are different types of historical movements. If time permits us, tomorrow we'll discuss about the historical movements. Why we need the cell culture means this is the area where the cell culture technology is currently playing a major role. There are model systems for studying the basic cell biotechnology, or sorry, basic cell biology, the interactions between the disease causing agents and the cells. Uh, Here it is the system which shows us the effects of drugs on the cells, the process and the triggering of aging and nutritional studies. And the other use is toxicity testing. Here the study that effects on the new drugs and the cancer research, obviously. So the study the function of various chemicals, virus and radiation to convert the normal cultured cells to cancerous cells. So the other one is virology, cultivation of virus for the vaccine production. Also use it to study their infectious cycle. And this is the technique. Cell culture is the technique which is used in genetic engineering for the production of commercial proteins, large scale production of viruses for use in the vaccine production like polio, rabies, chickenpox, hepatitis B, and measles. Gene therapy, cells having a functional gene that can be replaced to cells which are having non-functional genes. We already discussed about the tissue culture, in vitro cultivation of organs and tissues. Those were derived from the uh, organism after maintaining the temperature, incubator, supplemented media with a uh, nutrient containing growth factors is collectively known as tissue cultures. There are different types of cells grown in the culture is called connective tissues. Those are fibroblasts, skeletal tissue, cardiac tissue, epithelial tissue, liver, breast, skin, kidney, and many different types of tumor cells. So, as just we discussed about the primary culture and the continuous culture, also we discussed, and the types of cells. We will discuss this topic in the tomorrow's class. With this, uh,
Okay, ma'am.
thank you all. We'll continue the same topic tomorrow.
Thank you, ma'am.
Okay. I'm signing off.
Thank you, ma'am.
Okay. Thank you.